I am pleased to share the research highlights from the Division of Allergy and Immunology. We believe in sharing our ideas, our discoveries, and our advancements. The skin barrier is a promising target for prevention of allergic disease, though clinical trial results thus far have been mixed at best. Our faculty reported current and forthcoming skin barrier interventions that have clinically relevant implications in the prevention of allergic sensitization and atopic diseases. We research how enzymes that cleave proteins, called proteases, function in host defense and in the onset and propagation of inflammatory and allergic diseases. We focus on understanding how proteases and their inhibitors in the epithelium act in homeostatic and surveillance mechanisms and developing strategies for controlling proteolytic activity to treat diseases. Eczema is one of the most common skin disorders amongst children. The results of this study support certain immune cells, particularly CD4 positive T cells, responding to stimulation in an eczema-specific manner, resulting in disease and genotype-dependent changes in access to chromatin involving binding of the transcription factors, in particular NF-kappa B1. Asthma is the most common chronic condition in children, and it is the third leading cause of hospitalization in pediatrics. We developed a polygenic risk score for predicting increased risk of pediatric asthma across ancestries. Asthma exacerbations and severity have been linked to virus-induced secretions by dendritic cells and to eosinophils. We identified that type 2 inflammation regulates dendritic cell responses relevant to rhinovirus respiratory infection and eosinophilic airway disease, suggesting a potential mechanism by which eosinophil depleting therapies may reduce rhinovirus illness severity. We investigated the effect of vitamin D on esophageal inflammation. Vitamin D acted as a natural antagonist to the pro-inflammatory cytokine interleukin-13 and also regulated esophageal epithelial barrier functions. These findings suggest vitamin D is a therapeutic entry point for diseases involving type 2 immunity. Using immune liver cells, we have revealed cell-intrinsic and environmental effects of natural genetic variation on gene expression and demonstrated the use of enhancers as detectors of trans effects of genetic variation, providing a new resource for the understanding of the impact of genetic variation on gene expression. Through our sustained commitment to research and clinical excellence for allergic and immunological conditions, we will continue to change the outcome together. Thank you for your interest, generous research support, and inspiring patient and family participation.